Assalamualaikum warahmatullahi wabarakatuh. My name is Anisha Abdul Aziz, and today our group will be presenting about the calcium as signaling agent. First of all, let me introduce the rest of my group, which is consists of Brother Haikal Afiq, Sister Nur Rauda, and Sister Farsha Yunis. So first of all, what is calcium, right? So calcium ion or Cl2 plus are not only essential for building strong bones, but they also act as one of the most important and flexible messenger inside our cell. So all type of cells actually use calcium signals to respond to changes both inside the body and from the outside environment. So this calcium signaling process is actually tightly regulated by specific molecules, including proteins like calmodulin, Serca pumps, and calcium channel, as well as hormones such as parathyroid hormone and vitamin D. So this signal can differ in strength, timing, and patterns depending on what the cells need to do. So this precision allows calcium to play many roles, from regulating metabolism to triggering muscle contractions and even controlling gene expression. In today's presentation, we're going to focusing on the function, mechanism, health disorder, and also the scientific research about this topic.
Hello, Assalamualaikum everyone. My name is Nurul Rauda binti Fadil and matrix number 2414792. Okay, now I will be explaining about the functions of the calcium. One of the main things that calcium does is it helps in muscle contraction. When you want to move your arms, your brain will sense the signal through your nerve and into the cells, into the muscle cells. Then it will trigger the releasing of calcium. And then the calcium will bind to the special protein in the muscle cells that will lead to the muscle contraction. The next one, calcium also helps in the nervous system. It helps nerve to communicate with each other by triggering the releasing of chemicals called neurotransmitter. Another key function of calcium is that it can turn on the enzyme of protein inside the cell. When the level of calcium inside the cell increasing, it will bind to the certain protein. Then the protein will change the shape. Then it will activate the enzyme, which is the enzyme that controls metabolism and also cell division. Next one is calcium also involved in hormone release. For example, in pancreas, calcium helps in secretion of insulin, which is the hormones that control our blood sugar. Additionally, calcium involved in cell growth and also cell death. It is very important in immune system and cancer prevention. If the cell grow too much or don't die when they should, it can lead to the serious problems like tumors. Then, so calcium will regulate when the cell should divide and when it should shut down. Okay, that's all.
Hi, Assalamualaikum. My name is Muhammad Afiq, and now I would like to talk about the mechanism calcium in our body. So basically, mechanism calcium has entry and release. So calcium influx through voltage gated calcium channel, which is open in response to changes in membrane potential, and ligand gated calcium channel, which is open when an NMDA receptors bind. Then calcium will be released from endoplasmic reticulum through IP3 receptors and ryanodine receptors that in response to calcium influx. So the second one is calcium binding proteins. Calcium binds to sensor proteins like calmodulin to activate kinase or phosphatase, and bind to troponin C also to regulate contraction by actin myosin interaction in muscle cells. So the third one mechanism is temporal and spatial control. Basically, calcium signals are precisely regulated to trigger specific cellular responses. For example, through oscillations, repeated spikes in calcium ions allow cells to encode specific information such as T-cell activation, and through microdomains, localized calcium signals near organelles that can trigger specific responses without affecting the whole cell.
Assalamualaikum. I'm going to talk about disorder that related to our topic. First is cardiac arrhythmia. Under normal condition, calcium ion trigger the heart muscle to contract by entering the cells and causing more calcium to be released from internal stores. However, when the calcium channels like L-type channels or ryanodine receptor are damaged, this process becomes disrupted. Calcium might leak out at the wrong time or not be released when needed. This causes the heartbeat to become irregular, which is a condition known as arrhythmia. One specific example is a genetic disorder called CPVT, where stress can trigger the dangerous and sometimes deadly heart rhythm because of calcium leakage. Next is the Alzheimer's disease. In our brain, calcium is essential for nerve cells to communicate and form memories. In Alzheimer's disease, harmful substances like amyloid beta plaque and tau proteins interfere with calcium balance inside brain cells. Too much calcium builds up in the neurons, which activates disruptive enzymes like calpains and caspases. These enzymes begin to break down important parts of the cells, leading to neuron damage and memory loss. Last but not least, hypocalcemia and hypercalcemia. In normal condition, calcium stabilizes nerve and muscle activity. Hypocalcemia is a condition when calcium levels are too low. It can be caused by things like vitamin D deficiencies or problem with the parathyroid glands. In this case, nerve cells become overly sensitive. This can cause symptoms like muscle cramps and heart problems. On the other hand, if calcium levels are too high, known as hypercalcemia, this happens due to cancer or overactive parathyroid glands. High calcium makes nerves and muscles less responsive, leading to fatigue, confusion, and irregular heartbeat.
Now I'm going to explain about the scientific research that has been done by Professor Dr. Ebashi from Japan. He's the one that introduced how the actin-myosin related with enzyme which is calcium ions in muscle contraction. So basically, as you can see on figure 1, there's a calcium ion. We know that it's not gelatin agents like EDTA or EGTA. Basically, it directly regulates muscle contraction and relaxation. He demonstrated his experiment by removing calcium contamination and showing that even tiny amounts of calcium could trigger actin-myosin contraction. This proved that calcium role was real and physiological, not an artifact of lab chemicals. And then, as you can see on figure 2, what is mean the figure 2 tell us? Basically, it tell us about how the calcium ions can be measured, which is super precipitation. He identified sarcoplasmic reticulum binds calcium in an ATP-dependent manner, mimicking gelatin agents. He also demonstrated calcium ion-induced contraction leads to relaxation, forming the basis of dual regulatory role in muscle function.
Based on recent research, scientists found that too much calcium in brain cells can cause serious problems. It activates something called calpain, which acts like tiny seizures inside the cells. In Alzheimer's disease, calpain breaks the part of brain cells that help them talk to each other. It also helps form messy tangles inside the cells. Because of that, these cells stop working and die. This is why people with Alzheimer's start to forget things. In Parkinson's disease, calpain damages the brain cells that make dopamine. Dopamine is a chemical that helps control movement. Calpain also harms the cell's energy systems. As a result, the cells become weak and die, which leads to shaking, stiffness, and slow movement. In Huntington disease, there is a faulty protein called huntingtin. Calpain cuts this protein into pieces. These pieces stick together inside the cell and cause stress. Eventually, the cell dies. This leads to uncontrolled movements and thinking problems.
Why is calcium preferred as a secondary molecule over other ions? Well, it is because the levels of calcium in our cells is kept at very low levels. So even a small increase can lead to a strong signal that our body can detect. Okay, the next question is what could happen if calcium signaling goes wrong? If the level of calcium get too high or too low, it can lead to serious problems like muscle cramps, memory loss, or even a heart failure. That's all.
So scientists have made a lot of progress in understanding how calcium works in the liver, especially in normal function like metabolism and cell growth. So, however, these cases of acute liver injury, there's still a lot that we don't know actually. So, in recent years, many Western researchers still discussing about this, where the topic is ER cholinergic receptor induced calcium overload during liver injury. So one key problem here is the calcium will overload. So when liver cells get damaged, they release ATP, which activates P2X and P2Y cholinergic receptor. So this causes calcium to rush out the ER, which is endoplasmic reticulum, and also enter the cell from the outside. So when there's too much calcium in cytosol, it will overwhelms the mitochondria, which leads to cell damage and death. So to solve this, actually there is a potential treatment discussed by researchers where they suggest to combine ER calcium stabilizer, like drug that control the ryanodine receptors with cholinergic receptor blockers. So this combo, which is ER calcium stabilizer and cholinergic receptors blocker, actually could stop both the release of calcium from inside the cell and the flow of calcium from the outside. So as a result, it may help protect mitochondria and reduce liver damage. So more research is needed to test and develop this strategy actually, but I think it offers a promising direction for treating liver injuries and improving outcomes in liver disease and transplantation.
In conclusion, calcium plays a big important role as a signaling agent in our body. So let me relate this mechanism with Islamic perspective in Surah At-Tin. Laqad khalaqnal insana fi ahsani taqwim. That means we created man in the best of stature. This surah emphasizes the perfection in human design, which includes the precise regulatory system like calcium signaling. I think that's all from us. Thank you.